Right, so the nucleosome is the group of 8 histone proteins that DNA wraps around when it's compacted into chromatin. So blue being DNA, black being histone proteins. What's one of the roles of the nucleosome?
To condense.
So to condense DNA. What does this have to do with mutation?
Maybe it can't really be.
When you're really tightly wound, what?
You've had too much coffee.
Yeah. It might be less easy. If the DNA is really tightly packaged, it might be harder for some of those chemicals, like benzo pyrene, to access, to actually physically get to the DNA and mutate it. So you might expect that there's a relationship between how tightly your DNA is packed into chromatin and how many mutations occur in those parts of your chromosome. So that's why I came up with this second idea. Let's look at how nucleosome density might make a prediction. How does nucleosome density, how tightly packed your DNA is, affect the likelihood of mutation? So here's a different student example that was turned in. This one looks like on average about 1 mutation every 10,000 base pairs, so I liked this response. This is something I expected. I'm not saying other approaches are wrong, per se. But then when you look at nucleosome density, how do you interpret this plot, the gray dots? Which parts of this plot represent regions where the DNA is tightly compacted? What range of the chromosome in base pairs is the chromosome most tightly compacted?
On the left and right side.
The larger numbers, where nucleosomes are more dense. There are more of them per unit of chromosome. There's more protein, so these are more tightly compacted. So what would an expectation be of how many mutations would occur if our hypothesis is right, that how tightly packed your DNA is sort of protects it from mutation? What would you expect that? How would you expect that to have an effect on mutation? The dashed black line. Yeah, so you might expect there to be less mutation in those regions where it's tightly packed and more mutation, as this black dashed line that the student drew represents. So mutation rates you might expect to go up where the DNA is more accessible, where it's less tightly packed into protein structures. And all of this is meant to get at the question that was raised at the end of last class: are mutations random? Can we expect any sort of mutation to occur anywhere, anytime, on any part of any chromosome? An insertion mutation, a deletion mutation, a point mutation? Or can we detect biases? Is mutation non-random?
So when you talk about is mutation random, well, in relation to nucleosomes, where chromatin is inactive, it's not random there.
That's our hypothesis. So I'm not saying these are data. These are predictions that you're making about whether or not nucleosome, it's just I don't know. I didn't look at primary data. I just had this hypothesis. Well, maybe how tightly packed the chromosome is does have some sort of an effect on mutation. I'm sure people have looked at this. I just haven't researched it myself. So we've come up with a hypothesis. What would we do to test the hypothesis? Set up an experiment. So what would we have to do if we wanted to test the hypothesis there was a relationship, but we wanted to make a plot like this? What would we actually have to do in the laboratory? What? PCR. We'd have to be able to collect some sort of data on where nucleosomes are on a chromosome. How would you figure out where mutations are? We need to somehow identify the mutations. Normally that would be done with DNA sequencing. So you take a bunch of cells, you use some sort of technique to find out how dense the nucleosomes are, maybe some sort of microscopic technique, fluorescently label all of the proteins that are part of the nucleosomes, look at the chromosomes, measure how dense the nucleosomes are, how frequently those proteins occur along the length of a chromosome, and then, yeah, sequence the chromosomes, find out where the mutations occur. Are they evenly spaced, or are they more condensed in regions where you all happen to see less nucleosomes? And one of the overall learning objectives of the class is for you to identify experimental approaches to test hypotheses, to formulate hypotheses and to propose how to test them. So as we go through the class, I will be asking you more and more to do that, especially by the time the final rolls around. I'll tell you a story written on the exam. Tell me what sort of an experiment you think should be done. Another response. I liked this part. It looked like, yeah, every 10,000 base pairs, there was one mutation. And in between, there weren't any. That works for me. It's totally different looking than the other responses I've seen, but yeah, this represents what you might expect. Here is, to me, almost the ideal version. This is what I had in my mind when I wrote the question, but the problem was, and I apologize, I wrote the question ambiguously, which is why you see so many different types of responses. And to me, all of the responses I've shown do reflect student understanding of the principles and the concepts. Do you have any questions or concerns at this point about this particular topic? Again, this is brand new. We had never talked about these concepts in class, including nucleosomes. But a lot of you really stepped up to the plate and showed me that you understood what was going on, which was nice.
Okay, so the idea with the linear one, right, is the fact that there's approximately one mutation per 10,000 base pairs. So when you have 40,000 base pairs, there's approximately 4 mutations.
Right, so it looks like that was the plot that was made. Yeah. So that's basically totaling, as you move from left to right across the chromosome, how many total mutations have you observed to that point? That's totally fine. You could also do the moving average style, which is you look across 10,000 base pairs and you've seen one mutation. You look at the next 10,000, you see another mutation, and that's how those flat lines were generated. Students thinking, and that's how I was trying to word the question when I wrote it, but this line absolutely reflects the same sort of understanding. About on average, every 10,000 base pairs, you see a mutation. So this is what we're going to look for today when we look at real data from a published study that looked at mutation frequencies across a part of a chromosome. So now we have an expectation of what we think we're going to see if keep and going back. Okay.
So if that's a moving average, shouldn't the curve not go down? It should kind of flatten out.
So the bell curve,
Yeah.
that doesn't look like a moving average. That looks like maybe a plot of frequency of mutation. So in that case, you might say that the y-axis should not be it's not total number of mutations, it's mutation rate. So the highest likelihood of getting a mutation is here at the top of the curve where the nucleosomes are farthest apart. The DNA is most accessible. So if this was cumulative, goodness, let's see. You would see basically no mutation in areas of low nucleosome density. And then in there it would have a high frequency, steep slope. And then you'd also basically see no mutations in the other arm of the chromosome where there's high nucleosome density. That's what I would predict. I love graphing on Friday mornings, don't you? Is random. This thing?
The last one you showed with the.
One more back.
Oh no, yeah, one more back. Sorry.
Yeah, so I already told you what my prediction was.
The last one was about.
So
Can you go back to that one?
I would be from the hypothetical to the real. The point of talking about mutation in evolution, hopefully you already understand, is that this is where the arrival of the fittest occurs. We have to have mutations to get variation in the population that can be selected for or against. So when these mutations happen, one of two things will happen. Either mutations will be improving fitness, or they'll be neutral or decrease fitness. So if the mutation's beneficial, if it improves fitness, you'd expect that those individuals that have those mutations would be more successful, they'd have more offspring, and that allele would stick around longer over time, over numbers of generations in the population. That's the maintenance of the alleles in the population part. So this is critical. The idea that mutations are random is critical for evolution for a number of ways.
So you're saying they are random?
I'm saying, if I'm saying anything at all,
Oh, here we go.
I'm saying that it's critical for evolutionary biologists and for the theory of natural selection to demonstrate that mutations are more random than not random. So this is an example of some scientists trying to figure out, are mutations random? So we're going to test this idea. Are they random or not? This example uses benzopyrene. Charred hamburgers, delicious goodness. What they did was they looked at a sample of mutations, and where mutations in one gene occur, the p53 gene, it says right here, in lung cancer. So these scientists take a bunch of lung cancer cells from different patients, all of whom have mutations that are known to be in this one gene that leads to the development of lung cancer. So on the x-axis, they're plotting, like we did in our hypothetical plots, they're plotting relative position along the chromosome. In this case, it's codon number in this gene. So looking along the chromosome from left to right, position inside this p53 gene. What are they plotting on the y-axis? Number of mutations. In this case, it's in different individuals that have lung cancer. So you look at the same gene in a bunch of different people. Does mutation look like it's random or not? Are they evenly distributed all across the gene? Or are there peaks and valleys? There are definitely, well, I don't know about valleys. There are definitely some peaks, right? It looks like there are some particular regions of this p53 gene where mutations happen a lot more frequently than others.
So, I'm sorry, you're saying that this is population-based?
This is yes, this is population. The idea is this is population-based. It turns out that these are also areas, so that's what's plotted up here. It's real chromosomes from real people's lung cancers, looking at where in this gene do mutations happen. It's non-random. So then we have to wonder, well, why would it be non-random? Maybe it has something to do with where the nucleosomes are positioned in these genes, in these lung cancer patients.
And is that because it occurs more at certain codons than others?
So it's likely, it seems likely, yeah, why we're only looking in lung cancers. So what's probably true about mutations at these specific codons, at these particular amino acids in the p53 protein? So exactly, this is correlation between where does a mutation happen when you get cancer. So it's like these three amino acids are important in developing or preventing cancer, depending on which way you look at it. So whenever you look in a tumor, very frequently, when you see mutations in p53, it's at these three spots, and some others as well. Those are the tallest peaks. So So what Denisenko et al. In 1996 did was they took some normal cells, they grew them in flasks, and then they tossed some benzopyrene into the flask along with the cells. And they took the cells out and they asked where are the mutations that benzopyrene makes in these cells that are growing in dishes in a laboratory. That's what's plotted underneath the x-axis there. So these asterisks above 3 different nucleotides represent mutations that they saw in vitro in glass dishes or in tissue culture flasks. The presence of that mutagen making mutations in this gene. And look where they are. They're right underneath the peaks. Oops, wrong spot, right there. So in vitro, when you're just growing cells in dishes, when you add this known mutagen, benzopyrene, it makes, inside the flask, in the DNA of these cells, the same mutations that happen in real lung cancer patients out in the world. So is that correlation or is it causation? Does this prove that benzopyrenes are carcinogenic? Maybe. Does it prove that lung cancer is caused by inhalation of exhaust and other chemical and other particulate matter that has benzopyrenes?
It supports it.
It supports, yeah, it doesn't prove anything. It supports that interpretation. So this suggests that in this particular case anyway, that benzopyrene maybe makes non-random mutations, that it is selective about where it can attack DNA sequences and cause point mutations. The distribution of benzopyrene mutations here is non-random. It seems to be more likely to make certain mutations at certain parts of the gene.
Is there any significance to the fact that they're all G's?
Geez, I don't know. I will look into it because I didn't read this paper very recently, but I will look and see if they mentioned that. It is likely most mutagens have particular DNA bases that they will react with more frequently than others, so it might be most simply that benzopyrenes do react more with G's than A's, T's, and C's.
Okay.
But that's a great question, a good, well spotted. So all 3 of these sites where the benzopyrenes cause mutations were guanines. So to reiterate, if I'm saying anything at all, it's that evolutionary biologists would like to think that mutations are random. A lot of the time they are, but not all the time. So this is one case study to cite of non-random mutational processes. Sometimes mutations do occur repeatedly in the same spots in genes. It's not always totally random. And then you might ask, well, if I'm an evolutionary biologist, then why would I admit that mutations aren't always random? It's truth, and that's presumably what we're here to learn about. We've got another story to tell. This one's about frameshift mutations and whether or not certain types of mutations are good or bad. Can we make a rule about those sorts of things? We talked about this last class. I mentioned that it's true, and I'm happy to discuss this further if you want right now, that insertions and deletions can both lengthen and shorten proteins. So hopefully what we did last class will satisfy your thirst for knowledge about that topic if you were thirsty about it in the first place. But you can make insertion and deletion mutations that will do all those sorts of things to this protein. And a lot of times, I'm just trying to head off the normal question. At some point in this class, I guarantee you, before the end of the semester, if I don't talk about this now, one of you will ask this question. Can there be frame shifts that are beneficial? This always comes up. Not just frame shifts. Can there be point mutations that are beneficial? Can there be deletion mutations that are beneficial? Every sort of mutation you could think of, this always comes up. So tell me an example of how this works. So this is a relatively unknown example of this, but from a famous geneticist who you've never heard of, Susumu Ono. And this paper is from when? 1984. Proceedings of the National Academy of Sciences, bless you. This is a relatively famous paper by a relatively famous geneticist that you've never heard of. Sad for him. Okay, so here's the story. Back in the day, these plants would produce synthetic materials, polymers like nylon, and the effluent, the waste from the plants, would just dump out into waterways without regulation. And somehow, this is how science works, somehow somebody got the idea to go out into the rivers or streams surrounding these plants and collect bacteria, any sort of microorganism, and ask, can they survive in this sort of toxic environment with all these weird chemical byproducts in the water? And before Susumu Ono did this study, somebody else had previously published that there was a species of bacterium that could survive growing in nylon wastewater. And actually, they required, they could grow in media that only had this molecule as the only source of carbon. So normally we think about carbon sources for bacteria like you put glucose in the media, it's sugar, so that bacteria can eat it, metabolize it, and survive. This one species of bacteria that they found growing in these rivers could use this molecule as its food source, a byproduct of nylon production. Let me get a sense for how you feel about this. What's the probability? I mean, there are lots and lots and lots of bacteria out in the world. What's the probability that there was one species of bacterium that already had a gene that was necessary to digest nylon before the nylon production started? That is, was this like sticklebacks out in the ocean? Was there standing genetic variation? There was already bacteria out in the river that could eat this, and then it just happened that nylon production started, they started dumping this into the waters, and the bacteria were like, hey, cool, more food. Or do you think that it was evolution that enabled these bacteria to start taking advantage of this new resource? I hope I've sufficiently posed that question in a leading manner. And the title of the paper probably lets you know the direction this conversation's headed in. These bacteria evolved a new enzyme to digest this chemical. So as soon as the story is, as soon as this chemical showed up in the water, the bacteria that were living there was novelty evolved, something new, a new enzyme, a brand new enzyme that had never existed before on the planet, evolved in this particular species of bacterium. And that's what let them use this as their carbon source. This is the detail. How complicated do you think it is to make a new enzyme? Kind of complicated. Understatement of the week. Well, so why would it be complicated?
It has to fit they have to fit into each other?
Sure. So yeah, so you have to design a protein. How long are proteins in terms of number of amino acids usually? Yeah, like on the order of 80, 100, hundreds, 50 amino acids, 300 amino acids, 500 amino acids. So you have to put together a unique sequence of amino acids that has the right shape to bind to the thing it's trying to digest, and also has the catalytic activities that do the thing that it's trying to do. So it should be maybe relatively complicated. So what Susumu Ono did was he took that bacterium and he took other bacteria that didn't have the ability to make the enzyme necessary to digest nylon waste. and he found a single nucleotide insertion. One nucleotide was added to the genome of this bacterium, and that was all it took to take a bacterial species that couldn't digest nylon to one that could. One base pair. Way less work in terms of how evolution normally works. Way less work than you might predict. The addition of one nucleotide. That's what the black arrow is. So that T was added to the genome by chance, of course. This is natural selection. Random mutations happening during DNA replication in bacteria. One of them, the ancestor of all of these organisms that could digest nylon, had an extra T there. How does a single extra T help the bacteria leap to a new fitness peak? How does it make that radical transition to all of a sudden, hey, you've got a new enzyme? It's a frame shift. You've added one nucleotide. So that can create all sorts of havoc. It turns out that this frame shift was in an existing gene. So this is not in between genes. This is a frame shift mutation. The addition of a T was in an existing gene. So I'm going to underline the codons of the existing gene in red. So they were beforehand, let's see, I better double check my notes to make sure I'm doing this properly. I don't want to lead you astray at this point. So there was a CGA, there was a GAA, CGC, and each of those corresponds to the PRC line up here. That's the amino acid sequence of the PRC gene. That was the original gene that was there before this mutation happened. So there's a normal protein coding gene doing its thing. What happens when you add this T? After CGA, in the PRC gene, what's the next codon? TGA, a stop codon. So the first thing that this one base pair insertion did was make that original protein stop right there. So this mutation had one effect. It truncated a protein that was already being produced by the ancestral bacterium, which may or may not have something to do with the ability of these bacteria to digest nylon. So R2A was the new protein. So what else did this T do? Besides cause a stop codon in an existing gene, it also added ATG, a start codon. So then you start reading. So now this bacterium not only makes a shorter version of the PRC gene, but it starts to make a new gene from that start codon, ATG, AAC, GCA. So down here is the amino acid sequence of the newly born gene. Never existed before until a new T made a new start codon. And now you have a totally random series of amino acids. Of course, those nucleotides were always there. All of these nucleotides, they were already there. They were already encoding a different protein. But when you frame shift by one nucleotide and you make a start codon, you get a whole random new series of amino acids produced. Right? You can see this sequence of amino acids in the R2A gene has nothing to do with this sequence of amino acids that were produced on the gene that was essentially on top of it before that T was inserted. And so the R2A gene, that's how, in this case, those bacteria were able to, all of a sudden, one mutation, start digesting nylon. A whole new enzyme was born from one point mutation.
So is that truncated protein no longer functional, and then now that new protein that's created is functional?
That's the idea. That's the story that Susumu Ono told with this paper, was that the original protein stopped working. Presumably it would be deleterious to the bacterium. I don't remember offhand what PRC does, but R2A gave it a sufficient selective advantage to digest nylon, effluent, that it was overall net benefit. So a fabulous example of how evolution can happen really quickly, by chance. And of course, it was really, really, really only chance that the series of amino acids produced caused by this mutation happened to actually have some sort of useful effect for the bacteria. Other questions about this? One point mutation, not a point mutation, one insertion, single nucleotide, caused a stop codon and at the same time introduced a new start codon. So one more story. It helps us wrap up this review on molecular genetics. How mutations occur, how mutations can affect organisms. So this is a paper that just came out earlier this month, a week ago, I guess. And I saw it, I said, oh, I have to show the class this. Can synonymous mutations, what's a synonymous mutation again? Okay, so a mutation that doesn't change the amino acid sequence, can they be selected against or for? That is, can a synonymous mutation have any sort of selective benefit or cost at all? This is also a question that comes up a lot. So why are there so many synonymous mutations? Here's a great example of when we will see a synonymous mutation having an effect on an organism, which totally doesn't make sense. So here's what you need to know about this story to appreciate it. There's a protein in these bacteria called S20. It's part of the ribosome. Ribosome is the thing that translates RNA into protein. You might remember that ribosomes are composed of lots of proteins and lots of ribosomal RNA molecules. So these are big conglomerates of RNA and protein together, and that's what's shown here. So this is the 30S, the small ribosomal subunit. And one of its parts is the protein called S20, which is down here. And one of the roles of S20 is to stabilize the 16S RNA, which basically just means this protein interacts with RNAs that are part of the ribosome. Is the function of this protein important? This complex of proteins? What does the ribosome do? What does it do? Yeah, it makes proteins. It translates RNA into proteins. So you better be damn sure that if you screw up the ribosome, you're going to screw up the organism. If you can't make protein, you're dead. Okay. But it had been found, again, earlier than this paper, that there were certain mutations in that protein, the S20 protein. The one that these authors focused on is called A150C. Remember, that means that the 150th nucleotide is usually A, but this mutation makes it a C. That changes only nucleotide sequence. It's a synonymous mutation. That one point mutation doesn't change the the S20 protein sequence. But when a bacterium has just that one mutation, it severely affects the fitness of that bacterium. Synonymous mutation causes reduced messenger RNA and protein levels of S20. So if you don't have enough S20 messenger RNA and you don't make enough S20 protein, what's the effect on the cell? It doesn't form the ribosomes correctly because that S20 protein isn't there to be incorporated into the ribosome, so the cell doesn't make enough protein, so the cells are sick. Synonymous mutation in the S20 protein, in the S20 gene, causes cells not to form ribosomes properly, not to be able to synthesize proteins. How the hell would that work? Synonymous mutation doesn't change the protein sequence at all. It's actually pretty straightforward.
Is it like interactions with other proteins? Like they have to fold to each other and have like certain interactions between amino acids?
Right, so if the synonymous mutation, the question was, is it because of interactions between proteins? But this is the same as synonymous mutation. It didn't change the protein at all. What changed? The DNA sequence. So what does this imply? What are we to assume then about this synonymous mutation? Its effect is not in the protein. The effect is in which molecule? Either the DNA or the RNA. So synonymous mutation, when we talk about synonymous mutations, and oh, they must be neutral because they don't change the protein sequence. That's true. Synonymous mutations don't change the protein sequence, that's by definition. But they do affect the DNA sequence and the RNA sequence. So it must be the effect here on the ability of these bacteria to produce more proteins and form ribosomes must not have anything to do with the protein. It must have to do with the DNA or the RNA sequence.
What reads that are, the ribosomes are mostly made out of RNA molecules and some proteins.
And proteins, yeah. Ribosomes are made of RNAs and proteins.
So the ribosome isn't working, who's reading the proteins that go into the ribosome?
So if the ribosomes aren't working, then nothing's reading messenger RNAs. And so that's why there's no protein synthesis. So just to demonstrate with a little bit of data, these are 5 different mutants. WT is wild type. So 5 different bacterial mutants. And the one that we were talking about, the one mutation A150C is here, it's the 4th one. And what's being plotted here on the y-axis is relative exponential growth. So how quickly do the bacteria divide? You'll see wild type, strains that don't have any mutations have been defined to have a growth rate of 1. So the scientists have measured how quickly those bacteria divide, and they say that's a relative amount of 1. They're perfect. They're the pinnacle of bacterial life. And then relative to that, when you measure the growth rate of strains that just have that one mutation, A150C, they have like 60% of normal growth rate. So that one mutation does that. I'm only showing you this to prove, and that's why these authors put this in the paper, to prove that yes, this one mutation by itself does affect the fitness, natural selection, you would think, of these bacteria. Where's the A150 mutation? What's this? You might have seen figures like this before. What's true about RNA that's not true about DNA? Right, so RNA is single-stranded, but it's nucleic acid, so it wants to base pair when it can. And a lot of RNA molecules, especially messenger RNAs, will base pair with themselves, and they form these hairpin structures. So once a messenger RNA is produced, it tries to base pair with itself to stabilize itself. And that A150C mutation is there in the stem loop, in the stem of one of the hairpins. So it's not, these authors hypothesize, their conclusion is that it's not that this synonymous mutation, which it's true, it's a synonymous mutation, doesn't change the protein sequence, but it probably changes the stability of the messenger RNA's secondary structure. That is, this messenger RNA for this protein doesn't fold like this when it has this point mutation. It probably looks like something else. And if this messenger RNA is not stable, which it is in this structure, what might happen to that messenger RNA molecule that encodes the important protein? It could be degraded. So when messenger RNAs don't form these stable structures, maybe they don't get translated as much, maybe they get degraded by the cell more rapidly than normal. So take-home message, this point mutation doesn't affect the protein sequence, but maybe the hypothesis now is that it affects the messenger RNA's secondary structure that's formed after this gene is transcribed. So What's your take-home message? What's true about synonymous mutations?
They don't affect RNA or DNA.
Right, they don't affect the protein, but they still can have a selective benefit or cost. So just because it's a synonymous mutation, don't be fooled into thinking that it will be neutral, that it doesn't matter to the organism whether or not that mutation is there. Sometimes synonymous mutations really are important. So last thing about this. We talked about Rich Lenski's long-term evolution experiments. You put bacteria in a bunch of different flasks, you let them grow, you see how they evolve separately to tackle challenges. That's what these folks did in 2007. They took these bacteria that have this mutation, they don't grow very well. You saw that chart a couple slides ago. They do grow some, like 50 or 60% of wild types. They're okay, they're just not great. So they let those bacteria grow separately in different cultures over a long period of time to see how those bacteria might evolve, how random mutations might help some of them adapt to the fact that they've got this A150C mutation. These authors found a number of different ways, innovations, changes to the genome of the bacteria that helped them solve this problem. So what sort of things would you expect to happen in evolution by random mutation to fix the A150C problem? What sort of things could happen to that gene?
I mean, if it wasn't stable at that one spot, maybe make it stable somewhere else.
Okay, so you could have other mutations that help stabilize that hairpin in the presence of this mutation. You could have reversion mutations. That A150C could turn back from a C to an A. They didn't see that, as far as I understand. There are a lot of different ways that bacteria might compensate. One of the most striking patterns that they found was, well, let's see. What do you do when a ribosome, or when a cell isn't making enough protein to incorporate into all of its ribosomes? What do you need? So if your messenger RNA is unstable and you're not able to produce enough protein to satisfy all the needs of the ribosomes, what do you need? You need more ribosomes. You need more protein. So how does a cell crank up protein production?
More RNA, more protein,
So you make more RNA, you make more protein. How do you get more RNA?
more DNA.
More DNA. How do you get more copies of this gene? You have genome duplications, or parts of the chromosomes that contain that gene, even though it's got a mutation, right? One mutated version of this gene is sufficient to support like 50% of wild type growth. So wouldn't 2 copies of a half-functioning gene be better? And that's exactly what they found. That in multiple independent populations of this bacterial species that were growing in experimental evolution, in at least 4 of those populations, sections of the bacterial genome were duplicated. So in this case, these axes, these are radial plots. So the axis here is DNA copy number. So basically, if you're this far out from the radius, that's 1 copy. So if you're that far out from the center, that's 1 copy, 2 copies. Is that okay? So from the center, is how far out the plot goes. From the middle of each plot represents how many copies of that DNA are carried by a single bacterium. So for the most part, every strain has 1 copy of its DNA. But when this bumps out like that, that means there's been a duplication. Those genomes have 2 copies. And where in the genome, every time they found a more fit, evolved species of bacteria, what gene got duplicated as part of this random duplication? RPST, which is the gene that encodes that mutated gene. So the way that these bacteria solve the problem of having a dysfunctional gene, maybe the messenger RNA was being degraded too quickly, wasn't able to make enough protein to satisfy all the ribosomal needs for that ribosomal protein. These bacteria solve the problem by making more versions, more copies of that mutant gene, to make more messenger RNA, to make more protein, to get functional ribosomes. So in all, we've looked at mutations as a cause of variation, the origin of variation, arrival of the fittest. And we've looked at beneficial frameshift and a deleterious synonymous mutation. So we've got a couple of examples to talk about. And when we get together next time, there's nothing to do for the weekend. Come back Monday ready to talk about, well, there's nothing for this class, I'm sorry, to do this weekend. Come back Monday and we'll keep talking about evolution.